All right, welcome. So we're going to take a look at the neural conduction and synaptic transmission in this PowerPoint presentation. This is Dr. Mattson. So we're also going to look at how neurons send and receive signals. So we'll see if we can try and keep this, not too technical and get you all through this with ease. I'm going to try and keep this slide a little uncomplicated, but we'll see. When you look at like V and M, there's a definition of the electrical potential difference. And so V underscore N equals V underscore inside minus V underscore outside. And that is electrical potential difference is created by the separated charges across the lipid bilayer. So we're looking at the resting membrane potential, which is the steady voltage across a cell's membrane. So when the cell isn't sending a signal, and for most neurons, it sits around negative 70 MV, and so meaning the inside is negative relative to the outside. We measure this through an intracellular microelectrode. So fine glass electrode impales the cell. A reference sits outside, so we read it as the millivolts. There is things like, I guess, why the resting potential is important. Excitability is part of that. So it sets the starting line for action potentials. There's a driving force which determines how strongly ions move when channels open. We have in signal integration, and that influences the synaptic summation or the dendritic processing and timing. We also look at cell function and health, as to why this is important. And so that power is transporter as it affects cell volume. And we see this in excitable tissues like neurons, muscles, heart. It underlies contraction and rhythm as well. So we want to learn how the information is sent to and from the dendrites. And some of the neurons and how they interact with the terminals. Researchers study a neuron's membrane potential, or the difference in electrical charge between the inside and outside of the neuron. And so to record this membrane potential, the two electrodes are needed. So we have an intracellular electrode and an extracellular electrode. The intracellular electrodes must be the microelectrodes. They're usually made from saline-filled micropips by heating and pulling apart a fine glass tube with an electrical puller. The resting potential exists because positively and negatively charged ions are distributed unequally on the two sides of the neural membrane. So the concentration of Na+ is higher outside the neuron, and the concentration of K+ is higher inside the neuron. And so ions in solution are in random motion. The diffusion pressures, we look at the electrostatic pressures, like charges that repel and opposite charges attract. So the electrostatic pressure disperses any accumulation of positive or negative charges in an area. Ions pass through the cell membrane at special pores called ion channels. So when neurons are at rest, the membrane is resistant to the passage of the Na+ ions, only slightly resistant to the passage of K+ ions. Back in the 1950s, the neurophysiologists that we have, Hodgkin and Huxley, provided the first evidence that an energy-consuming process is involved in resting potential. So active mechanisms in the neural membrane continuously transfer Na+ ions out of the neuron and K+ ions into the neuron. This occurs at a ratio of 3 Na+ ions moved for every 2 K+ ions moved in. So there's a loss of 1 positive charge every time the pump is activated. So the extracellular fluid contains several sodium ions and a few potassium ones as well. The cytoplasma contains more potassium than sodium ions, and the 3 scenarios of ion movement that we see illustrated with the arrows across the cell membrane. So the first ions in motion move down their concentration gradients. So the sodium ions will tend to enter, and then the potassium ions will tend to exit. The negative interval charge creates pressure for both the sodium and potassium ions to enter. And then the sodium-potassium pumps, indicated as the blue circles, on the cell membrane transport 3 sodium ions out for every 2 potassium ions that they transport in. So now that hopefully you have a better understanding of the state of resting neurons, we're going to take a look at how electrical signals are created in a resting neuron. The postsynaptic potentials are changes in the membrane potential produced by the action of the neurotransmitters released by the presynaptic neurons. When we look at the excitatory postsynaptic potentials, or EPSPS for short, these are depolarizations, and they increase the likelihood that a neuron will fire. The inhibitory postsynaptic potentials, or the IPSPS, are hyperpolarizations, and they decrease the likelihood that a neuron will fire. The postsynaptic potentials have 3 important properties. The first one is that they're graded. Their amplitude is proportional to the intensity of the input. That is, stronger stimuli produced bigger EPSPs and IPSPs. They are transmitted decrementally, and so as they passively spread from their site of generation, they get weaker as they go, kind of like sound does through air. And then the third point is that they're transmitted rapidly. So like electricity through a cable, so rapidly that transmission is usually regarded as being instantaneous. Action potentials are generated at the axon hillock, and an AP occurs when a neuron is depolarized to the point that the membrane potential at the axon hillock reaches about 70 MV, the threshold of the excitation for many neurons. Unlike the EPSPS and the IPSPS, APs, action potentials, are not graded. They're all or none, so full-blown or not at all. These are 2 kinds of neural integration for the multiple signals received. We have the spatial summation, which can involve 2 or more EPSPS, 2 or more IPSPS, or we could see both EPSPS and IPSPS. And then the temporal summation, which can involve 2 or more EPSPSs or 2 or more IPSPSs. And then the EPSPS and the IPSPS cannot temporarily summate due to the fact that just a single synapse is involved, and it must be either excitatory or inhibitory, but not both. When the threshold of excitation, which is about -70 MVs, is reached, voltage-gated Na plus channels open momentarily, and then the Na plus ions rush into the neuron under tremendous pressure from their concentration and the electrostatic gradients. This drives the membrane potential to about a positive 50 millivolts. At the same time, the voltage-gated K plus channels slowly begin to open, and most of these channels open at about the time the membrane potential is about positive 50 millivolts, and then at this point the K plus ions are driven out by the positive 50 millivolt charge, and then their high internal concentration repolarizes the neuron. It leaves it slightly hyperpolarized for a few milliseconds. Because only a few ions that are adjacent to the membrane are involved in the generation of an action potential, the resting potential is readily reestablished by the random motion of ions, not the Na plus K plus pump. The line graph here shows the steps and phases in an action potential. And the line graph has time in milliseconds, ranging from 0 to 5 on the x-axis, and the membrane potential in millivolts, ranging from negative 90 to a positive 60 on the y-axis. The graph plots a line with a single upward peak, whose components can be described in terms of the time frame. So 0 to 1 millisecond That's a flat line at -70 millivolts. And the sodium channels open up at around 1 millisecond. When we look at the 1 to 1.4 millisecond, there's a steep upward curve that peaks at around 50 millivolts. This upward climb is labeled rising phase, and the potassium channels open during this time. The 1.4 millisecond to the 2.2 milliseconds, this is at the peak of the 50 millivolts. The sodium channels close, and there is a steep downward curve ending at -70 when potassium channels start to close. This phase is called the repolarization. The 2.2 to 4.5 milliseconds, this is the downward curve. It very slowly goes downward, still reaching around a -86 at 3 milliseconds, and then slowly starts to climb up, reaching a -70 at around 4.5 milliseconds. And then this phase is called the hyperpolarization. And then the last one is the 4.5 to 5 milliseconds, and then the line is flat at 70 millivolts after that. For 1 to 2 milliseconds after the generation of an action potential, another action potential cannot be generated no matter what kind of input the neuron receives. And so this time period is called the absolute refractory period. This period is then followed by the relative refractory period, during which an action potential can only be elicited by high levels of stimulation. Once an AP is generated, the action potential is generated at the axon initial segment. It's transmitted along the axon. The purpose of axons is to transmit APs from the soma to the terminal buttons of the neuron. Conduction of the APs along an axon is not like the transmission of PSPS. The conduction of the EPSPS and the IPSPS is passive, like the electricity through a cable. So it's instantaneous and decremental, whereas conduction of an AP along an axon is active and therefore slower. When voltage gated at the Na positive channels on the Hillcock membrane, the Na plus ions rush in and a full-blown AP is then generated. The electrical disturbance that is created is transmitted passively to the next Na plus channels along the axon in response. The voltage gated Na plus channels therefore open and another full-blown potential is generated. But in reality, the sodium channels are so tightly packed that it's best to think of APs as waves of depolarization spreading down an axon. The AP is then regenerated along the length of the axon, and the APs also spread from the hillock back through the cell body and the dendrites. And there's not a lot right now that's currently known about the functions of these potentials. Transmission in the normal direction from the hillock to the terminal buttons is called the orthodromic conduction. However, if the terminals are electrically stimulated, the APs can be generated and actively transmitted back to the hillock. This is called the antidromic conduction. Many axons are myelinated by the glia cells, and myelination insulates the semi-permeable axon membrane, blocking the flow of ions through the axon at all but the nodes of the Ranvier. This actually improves transmission. In myelinated axons, APs travel passively between the nodes of the Ranvier, but at each node there's a pause while a full-blown AP is generated. This is called the saltatory conduction. Saltatory means to skip or to jump. Because much of the transmission of the APs in myelinated axons is passive from node to node, transmission in myelinated axons is faster and it requires less energy. Larger axons conduct faster, myelinated axons conduct faster. Many interneurons do not have axons and their transmission is decremental. The image here illustrates the direction of signals that travel through a neuron using arrows. And so the signals start at the dendrites, they move down toward the axon, they travel through an axon as a single unit. At the base of the axon, it branches out into each of the terminal buttons. So we have the postsynaptic potentials, which is the PSPs. These are elicited on the cell body in the dendrites. The PSPs are conducted decrementally to the axon, and when the summated PSPs exceed the threshold of excitation at the axon, an action potential or AP is triggered. The AP is conducted non-decrementally down the axon to the terminal button, and then we have the arrival of the AP at the terminal button, and that triggers the exocytosis. So a few slides ago, you heard me mention Hodgkin and Huxley as the researchers. So their model, traditional model, is too simplistic to fully represent the neural conduction. And so the original model does not account for the many neurons that fire APs in the absence of input or don't fire APs at all. The APs vary in terms of amplitude or duration and frequency. And then the dendritic function is more complex than previously believed. So the dendrites, like for example, would appear to be able to actively conduct axon potentials. Some synapses occur on little buds on dendrites. These buds are called the dendritic spines, and other dendritic synapses occur right on the dendrite shaft. So in addition to the axosomatic and the axodendritic synapses, we have the axoaxonic synapse, the dendrodendric synapse, the dendroaxonic synapse. These are the synapses between the main shafts of axons, and then we have the non-directed synapses. Some of the dendritic synapses can transmit in either direction. The axoaxonic synapses mediate the presynaptic inhibition or the postsynaptic inhibition, which is mediated by the axodendritic and the axosomatic synapses. Most synapses that are discussed in the textbooks that you're reading are the directed synapses. And these are synapses where the site of release and the target site are close in apposition. Then there are also the non-directed synapses. For example, some presynaptic axons have a string of beads appearance, and the neurotransmitter is widely dispersed from each bead to many targets in the general area. This arrangement is common for the monoamine neurotransmitters. The image here shows a neuron with its axon branching out at its end, and an inset image in this picture shows a magnified view of one of the branches, and each branch has spherical bulges at regular intervals, and then each bulge has small spherical structure called the veracosities, and each veracosity contains several neurotransmitter molecules, which have also been released outside the neuron. There are two main types of neurotransmitters. We've got the small molecule transmitters, which is synthesized in the cytoplasm and packed into vesicles by the Golgi complex, and large molecule transmitters that are synthesized in the soma by the ribosome and then moved down to the terminals by the microtubules. And there can be small and large molecule transmitters in the same terminal button, and this is called coexistence. The arrival of an AP or action potential at a terminal button opens voltage-gated calcium channels in the button membrane, and then the Ca plus ions enter the button. The entry of the Ca plus ions cause the synaptic vesicles to fuse with the presynaptic membrane and empty their contents into the synaptic cleft, which is a process that we call exocytosis. Small molecule transmitters are usually released each time an action potential arrives at the terminal. By contrast, we have the large molecule transmitters that are released gradually in response to multiple action potentials. Most documented cases of coexistence have involved one small molecule neurotransmitter and one neuropeptide, although the coexistence of two small molecule transmitters has also been noted. We look at the metabotropic and the ionotropic receptor. Both of these when we look at binding of the neurotransmitter to its receptors, it can influence the postsynaptic neuron in one of these two ways. And so the metabotropic receptor associated with the signal protein is attached to the G protein inside the neuron. So the G protein can either move to a nearby ion channel, activate it, and cause a change in the membrane potential, or it can lead to the production of chemicals called second messengers. That can have more enduring and far-reaching effects on the sensitivity of the neuron. And if it's the ionotropic receptor, this is directly associated with a channel in the postsynaptic membrane and can induce brief EPSPS or IPSPS. So after it's released, the neurotransmitters diffuse across the synaptic cleft to the postsynaptic membrane, and that's where it binds to the receptors in the postsynaptic membrane. And then the specific receptors for each of the neurotransmitters is what I just covered. Neurotransmitters are deactivated in the synapses by one of two mechanisms. Some neurotransmitters are broken down in the synapse by enzymes, and other neurotransmitters are deactivated by the reuptake into the presynaptic neuron, where they are then recycled. So the process of this, two mechanisms in neurotransmitter deactivation in synapses is this. The reuptake, which is the image that shows a neurotransmitter molecule being released from the tip of a button, reaching a receptor on the surface of the postsynaptic membrane and going into the bubble again through a channel labeled as the transporter in the presynaptic membrane. And then the other one on the right is the enzymatic degradation. And this image shows a neurotransmitter molecule being released from the tip of a button, and it reaches the synapse when it comes into contact with deactivating enzymes, shown as thin strands. And after contact, the neurotransmitter breaks into pieces. The gap junctions are narrow spaces between adjacent neurons that are bridged by fine tubular channels that contain the cytoplasm. So the cytoplasm is continuous, allowing electrical signals and small molecules to pass from one neuron to the next, and sometimes they're called electrical synapses. These gap junctions allow direct communication between two or more neurons in the nervous system, and these types of connections seem to be an integral feature for the local inhibitory circuits. The gap junctions can also form between the glial cells or between the glia and the neurons. The existence of these gap junctions between pre- and postsynaptic neurons and adjacent astrocytes can form the tripartite synapses, allowing the astrocytes to synchronize actively between groups of neurons. So the image here shows the cell membranes of two adjacent cells, and each membrane is a lipid bilayer with the strands of the layers facing each other and the spherical molecules on the other side. The layer facing the other cell is labeled the pre-junction membrane, and the other layer is labeled the post-junction membrane. So we have several cylindrical pores labeled the connexins, which connect the cytoplasm of the two cells. So we have the amino acid neurotransmitters, and these are the individual building blocks of proteins. They also serve as the neurotransmitters at fast-acting or point-to-point synapses. There's a conclusive evidence that glutamate, aspartate, glycine, and the GABA are neurotransmitters. And the monoamine transmitters are often released from a string of beads or a string of bead-type axons, and they have slow, lingering, and diffuse effects. These are neurons that release that typically have their cell bodies in the brain stem. Acetylcholine is a small molecule transmitter at the neuromuscular junctions. And so many synapses in the ANS and even some at the CNS synapses, it's created by an addition of the acetyl group to a choline molecule. Acetylcholine is the only neurotransmitter known to be deactivated in the synapse by the enzymatic degradation rather than by the reuptake. it's deactivated. And with the unconventional neurotransmitter, we look at soluble gas neurotransmitters like nitric oxide and carbon monoxide. These gases are produced in the neural cytoplasm and diffuse immediately through the cell membrane into the extracellular fluid and into nearby cells. And this stimulates production of the second messengers. They're difficult to study as they act rapidly and they're immediately broken down, existing only for a few seconds. The anandamide is an inhibitory kind of cannabinoid, marijuana-like neurotransmitter, and it's released from dendrites as well as cell bodies. Neuropeptides, the peptides are short chains of 10 or fewer amino acids. So over 50 peptides are the putative neurotransmitters, and they are the largest neurotransmitters. There's 5 categories of the neuropeptides, and they all have widely varied functions. Endorphins are an example of a neuropeptide transmitter. They're more opiate-like transmitters. They're important for things that relate to the reward system of the brain. You've heard me mention a few times a string of beads, and so this image, shows a string of beads. It shows a highly interconnected, bright fluorescent web. The background is almost totally black with traces of yellow, and throughout the web are bright beads at small intervals. So drugs that facilitate a transmitter's effects are called the agonists, and drugs that reduce a transmitter's effects are called the antagonists. And drugs act upon one or more of the 7 steps here in the neurotransmitter action, and the exact mechanism varies from one drug to the next. The image here shows the formation of the neurotransmitter. These are the 7 steps in the neurotransmitter. So the first one is the neurotransmitter molecules, and these are synthesized from the precursors under the influence of enzymes. The synthesizing enzymes, which are illustrated like springs, act on neurotransmitter precursors, which kind of look like quarter spheres. The enzymes convert the precursors to form neurotransmitters, which are illustrated as spheres. The second one, the neurotransmitter molecules are stored in vesicles. The vesicle is also spherical with an outer membrane, and each is seen to contain 3 neurotransmitters. The third one is the neurotransmitter molecules that leak from their vessels are destroyed by degrading enzymes. The fourth one is action potentials cause vesicles to fuse with presynaptic membrane. An opening toward the synapse is created, and then neurotransmitter molecules are released into the synapse. Number 5, the released neurotransmitter molecules bind with autoreceptors in the presynaptic membrane and inhibit subsequent neurotransmitter release. Number 6, the released neurotransmitter molecules bind to the postsynaptic receptors in the postsynaptic membrane. And the last of the 7, or the steps, number 7, the released neurotransmitter molecules are then deactivated by either reuptake, where the molecule goes back into the bubble, or the enzymatic degradation. So when we're looking at how drugs have been used to influence neurotransmission, we look at different receptor subtypes. And so acetylcholine binds the 2 main types of receptors, and the 2 receptor types have a different distribution in the nervous system, and then they're going to interact with drugs differently. With the discovery of the opioids, the drug morphine, we would look at like for pleasure and pain. This would bind to the opioid receptors in the brain. And when looking at the antipsychotic drugs, we might look at like tremors and mental illness. So the first 2 drugs that effectively treated some of the symptoms of schizophrenia were later found to be dopamine D2 receptors or antagonists. And this is going to wrap up this PowerPoint for today. If you have any questions, feel free to reach out. And I hope this helped to some extent explain a little bit more of the neurons and neurotransmitters.